final roundtable and discussion for today is looking at how patient care will be changed with microbiome medicine, especially when treating serious cancers, serious oncology, and infectious diseases. From Seventure to moderate this panel, it's a pleasure now to hand over to Isabelle de Cremoux. Isabelle. Is it working? Would you like to try another one?
No, that's okay.
Good. Okay.
Good evening. Very, very happy to be here with you and be recorded, and with the people that are attending digitally for this last session of the day, but last but not least, for a super interesting theme and a highly qualified panel. I have the privilege and the pleasure to welcome tonight for our session, digitally connected, Professor Hassane Zarour from Pittsburgh. Is there any need to present Professor Hassane Zarour, one of the eminent professor in immunology and dermatology, having many roles including melanoma research and a lot of other indications around cancer and skin cancer specifically. Here in Paris, on my left, another person that is not really needed to present, Professor Mohty, specialist of bone marrow transplantation and microbiome role in hospital setting, and especially in Lyon, as well as in INSERM, and in a lot of associations such as the European Society for Blood and Marrow Transplantation, where Mohamad was the previous president, and currently the Acute Leukemia Working Party of the EBMT and the International Academy for Clinical Hematology. Then, two entrepreneurs, Hervé Affagard, co-founder and CEO of MaaT Pharma, based in Lyon, who will present his activity, so I don't present the company. And Tim from Microbiotica. I have trouble pronouncing your name, so that's why I'm calling Tim. Tim Sharpington, is that correctly pronounced? Okay, good. So CEO of Microbiotica, a microbiome company based in the UK. And to complete this panel, we are more than pleased that Ferring had enough time and bandwidth to actually send us a speaker in the name of Bilbo, Senior Vice President and Global Head of Microbiome Franchise at Ferring. So without delay, we will start with Professor Mohty's introduction on how useful is microbiome for a serious case of hospitalized patients in his own field.
Thank you very much, Isabelle, for the kind introduction. Good evening, good afternoon, everybody. It's a true pleasure to be with you today, and I think I have maybe 2 or 3 minutes to try to convince you that modulation of the microbiome or microbiota is definitely going to be the next pillar of cancer therapy in general, but also many other health conditions or diseases. I thought, for the sake of introduction, to kindly remind this audience about the pillars of cancer care and the evolution over the last almost 100 years, because the story started with radiotherapy, and in the 50s and 60s we had the advent of conventional chemotherapy, and I'm sure everybody's familiar with this, unfortunately, because I think every family somewhere has seen a case, a neighbor, a family member, or whoever who is affected by cancer. And these conventional chemotherapies will continue to be an important tool for treatment. Then in the mid-80s, 90s, late 90s, we've seen a true revolution with targeted therapies, small molecules. The best examples are the monoclonal antibody, for instance, anti-CD20 rituximab, but also imatinib, which has been a true revolution in terms of targeted therapy. But now, in the last decade, I think it is a revolution of immune therapy. And under the umbrella of immune therapy, you do have several chapters, I would say: vaccines, checkpoint inhibitors, adoptive cellular therapy, bispecific antibodies, T-cell engagers, CAR T-cells, but many other tools to come. So if we put together in sort of what I mentioned, the pillar, I think today in 2022, 2023, we do have a strong body of evidence, and you will hear about this in a few minutes, to highlight that today modulation of the microbiota is really the next pillar, the next frontier. So we are adding an additional tool to cure more and more patients. And why is this important? Why I am a strong believer that this is going to happen and this is going to be a new frontier? Because actually, we have been we neglected the role of the microbiota in the human physiology, whereas I think everybody is well aware of what's ongoing there in our gut. And, you know, whenever you've got a diarrhea, you won't feel very well. So infections are really the very good example of what can happen to the microbiota. But actually, when you disrupt the microbiota, this is creating an unhealthy condition, an abnormal condition called dysbiosis. And this is a true big problem that you can see in many cancers, but also inflammatory and autoimmune conditions. And unfortunately, this dysbiosis is not only has probably a causal role, but also is also contributing to fuel the fire. And interestingly, the treatments we use in cancer by themselves, like chemotherapy, radiotherapy, but also the broad spectrum antibiotics, are further fueling the fire. So they are even aggravating the dysbiosis. So you would guess it's a sort of a vicious circle, and at the end of the day, it's about developing tools, treatments, drugs to correct, to cure this dysbiosis. And in all our interventions tomorrow, I think in health conditions, we will need this microbiota modulation. Thank you very much.
We'll then go to Pittsburgh to hear Professor Hassane Zarour.
Okay, so thank you very much. Thank you for your introduction. Thank you, MaaT, for this great introduction to underlining the role of the microbiome in cancer therapy. So I'm professor at the University of Pittsburgh. I'm a cancer immunologist interested by approach to improve treatment of patients with cancer, in particular skin cancer and melanoma. And I've been recently very interested in the role of the microbiome. And I would like to review with you evidence supporting the role of the microbiome in cancer immunotherapy, both preclinically and clinically. So this, I would like to start with underlining preclinical evidence supporting the role of the microbiome in regulating, modulating effect of cancer therapy. So I might just talk about that. And this preclinical model, in mice model many years ago, evidence and interesting publications supporting that certain microbes in the gut play a critical role in regulating the efficacy of classical chemotherapy to treat cancer. And several drugs, oxaliplatin, cisplatin, cyclophosphamide, that are being used in patients with cancer in this very interesting, I'm not going to go into much detail, but this model really showed that certain microbes that have been identified play a critical role in modulating the effect of this chemotherapy in this mice model, modulating their anti-tumor activity. And in this very mechanistic paper, pin down the mechanism used by the microbes to mediate the effect of chemotherapy. But most interestingly, following this evidence with chemotherapy, additional work that this was not only true for chemotherapy, this was also true for immunotherapy. And two seminal papers several years ago showed that some microbes were critical to regulate the efficacy of drugs that are now FDA approved and efficacious in patients with cancer, like anti-CTLA-4, and this work of Laurence Vogel in Paris, or anti-PD-1, and this was in mouse model showed by Dr. Gaevski group in Chicago. So, as you know, immune checkpoint blockade and immunotherapy is one of the most potent treatment of cancer. This is true in melanoma, where advanced melanoma benefit from this drug. 30 to 40% of patients with advanced melanoma are cured or do very good upon treatment with this immune checkpoint blocker, either anti-CTLA-4 or anti-PD-1 or both. And we also know that this is true for other cancer. Many cancer benefits from immunotherapy, anti-PD-1, anti-CTLA-4, or both, either alone or in combination with chemotherapy. I think definitively immunotherapy in particular, this antibody targeting this inhibitor receptor, anti-PD-1 and anti-CTLA-4, have been definitely one of the most potent treatments of cancer. So, and as I said before, based on this preclinical model, the evidence was brought that the microbiome play a critical role in mediating this effect of this immunotherapy. And in fact, following the mice data, a number of studies have shown that gut microbiome composition or certain microbial signature in the gut correlated with response with immune checkpoint blockade. Again, there were a number of similar papers published 5 years ago in melanoma, lung, and renal, showing that certain increased abundance of certain microbes directly correlated with the likelihood of response to immune checkpoint blockade. And so some microbes have been identified. They are listed here. I'm not going into much detail. And since then, many other studies have confirmed this same evidence, this correlation between gut microbiome signature and positive outcome upon treatment with immune checkpoint blockade. So this was great, and I think following the mouse model, this was another set of evidence supporting that we could possibly use the microbiome to the benefit of the patient, and I think that's really opened the door to a microbiome-based treatment of cancer. One down point, and I think one concern was in this study, although the principle was there, is the lack of commonality between the study, and there was some significant difference in terms of what is a good microbial signature associated with response. And I think this is the topic of very active research in the field. Okay, yeah, so this is our contribution to the field, where we really did the same study in melanoma patient with advanced melanoma, looking at the gut microbiome composition of this patient, either pre or on treatment, and correlated with outcome, and could confirm that this is a complex figure, but if you look at the gut microbiome pre-treatment, this correlates with outcome, good outcome. And I think the most important parameter is the fact that patients do not progress after a year. So it's still unclear why it is the outcome at 1 year and not before, maybe supporting, and that's an area of investigation, that the role of the gut microbiome may be more important later on during disease progression, that maybe during the first year of treatment. Other mechanism, maybe mechanism within the tumor microenvironment may be more important to drive response or resistance to treatment. And for some reason, the gut microbiome may play a role more importantly down the road. So this is also something we are investigating. But overall, when we look at what was the signature associated with response in a cohort of patients, we found again a lot of similarity with what have been published in terms of taxonomic identification of bugs associated with response, of not response. So really supporting the relevance of the finding. So I just want to underline here that although we focus on the gut microbiome composition and response or non-response to treatment, it is also important to say there is also strong evidence, and that's what we also have published, that gut microbiome composition is also and correlates with occurrence of adverse event. And as you know, this is an important clinical concern. Many patients, although they benefit from immune checkpoint blockade, have sometimes to stop treatment because they have significant adverse events. So the possibility to use a microbiome not only to improve clinical response, but to decrease or prevent the occurrence of adverse events will be critical to allow this patient to be treated by treatment they benefit from. And this, in this slide, we definitively identify a gut microbiome associated with occurrence of adverse event. And we also show that the gut microbiome associated with adverse event correspond to what is a good gut microbiome in terms of clinical response. So this is something we need to keep in mind using gut microbiome modulation or approach, not only to improve clinical outcome, but to decrease the occurrence of adverse event and allow patients to be treated for a long time with potent immunotherapy of cancer. We have tried and to have a better understanding of the mechanism, and this is also a very active area of research, both preclinically in mouse model and in humans. So if the microbiome is beneficial to patient, what are the mechanisms? So it's still a very early of year research, and there's no strong answer or in this area. We have tried to pin down the mechanism, and the way we did that is in the stool of patients who respond or not respond to immunotherapy is to look at the host cells. So we disregard the microbes and look at the host cells. So what cells from the host who are present in the stool of responder and non-responder, and we evaluated transcriptomically, so with RNA evaluation, the gene profile of these cells. And we make two important findings, I think, which are food for thought. The first is in the non-responder, we found that the host cells were predominantly inflammatory cells, immune cells, while in the responder, they were mostly epithelial cells. So more inflammation in the stool of the non-responder as compared to the responder. And when we evaluate the gene pathway and based on the transcriptomic analysis, this analysis pinned down to chronic inflammation, a number of genes like LPS and IL-8 or IL-1 beta, showing that there was evidence of chronic inflammation in the stool of the non-responder, donc supporting that LPS activation and chronic inflammation may play a critical role in driving non-response to treatment, and that possibly certain microbe may play a critical role in driving this chronic inflammation and may represent an impediment to response to immunotherapy. Before concluding, I would like to underline that beside the work to really try to understand the role of the microbiome in regulating response to immunotherapy, there was also, I think, at this several years ago, at least from us, the need to show that we could manipulate the gut microbiome to provide benefit to patients. So And we did that in designing a clinical trial that was published a year or so ago, a fecal transplant. So you know the fecal transplant, microbiota transplant is used, in certain disease, and it's pretty safe. So we wanted to show if the gut microbiome can be beneficial to patients, if we could convert non-responder to anti-PD-1 to responder patient. So we designed this trial where patients were treated with anti-PD-1, advanced patients with stage 4, so very advanced melanoma, with a response to PD-1. And when they didn't respond, to treat them with one single administration of fecal microbiota transplant and again anti-PD-1. And this fecal microbiota transplant was obtained from long-term responders, so people who were treated with anti-PD-1 and with extremely good, either were total responder, complete responder, or very long-term responder. So, and to show some type of proof of principle, and we published that a year or so ago. And the good news is in the 15 evaluated patients, 6 patients had a good outcome. So 6 had objective response with significant shrinkage of the tumor burden. Other one had a prolonged stable disease. And again, these were patients who did not respond to anti-PD-1 alone. So I think, and in the context of this trial, I'm not going to go into detail, we show that we could not only provide a benefit to patients, but have evidence that we were capable of modulating their immune response to cancer, both in the periphery and in the tumor, showing really, demonstrating that microbiome-based therapy could be used to the benefit of the patient and increase the likelihood of response to immunotherapy of cancer. I would just underline that while we are doing the study, a similar study was done independently by a group in Israel. It was a smaller trial with 10 patients, but the findings were similar, and they found that 3 out of the patients did respond to FMT and anti-PD-1. And I think this has infused a lot of enthusiasm in the field. I think justified enthusiasm in the field that we can really work on developing a potent microbiome-based treatment of cancer, which maybe in the end may not be fecal transplant, maybe pills with stool, or pills with a cocktail of microbes once we have a better understanding of the composition and what microbes need to be in the pill. So I'm going to conclude here this brief talk. This is my last slide. Yeah, sorry for the delay in the slide. So I think that microbiome therapy have opened a new horizon to improve outcome for cancer patients. So we have reviewed the data supporting that gut microbiome composition pre-treatment correlates not only with the outcome, good and bad outcome, but also to adverse events. We also review that there is evidence that we can improve the outcome of patients with microbiome-based therapy of cancer. This has been shown in this early trial with fecal transplant, and it's a very active area of research. Many other ongoing trials are trying to reproduce the data with fecal transplant in melanoma and other tumor. Other groups are using different microbiome-based treatment, pills, cocktail. So we are looking forward to know more about this trial in the future. I briefly talk about that, but I just want also to mention that there is also clinical evidence, a very small trial, but still strong evidence that you can use the microbiome-based treatment to decrease the occurrence of adverse events in the patient. So I'm going to end up here and give you back the...
Thank you. This is one thing that I really love when investing in the microbiome field. It starts from patients in real life, real hospital with real physicians, observing the mechanism of action. And even if there are still assumptions to be validated, we move from the patient to the products and to the lab and to the validation and to the innovation. And that's why we started by 2 hospital physicians. Now, turning to the entrepreneurs, you will have illustration of how entrepreneurs can develop products aiming at regulatory approval and high market sales, starting with MaaT Pharma and hematological malignancy with Hervé Affagard.
So, yeah, thank you. Thank you very much, Isabelle, for the introduction. So indeed, I'm the first illustration in terms of the application. So I'm honored to be following Professor Mohamad Mohty and Hassane Zarour. So I'm Hervé Affagard. I'm the founder and CEO of MaaT Pharma, and I'm also the president of the Alliance Promotion Microbiotica, which is a French association promoting the leadership of France in terms of the microbiome translation. Just trying to see. Yeah. So we are a listed company, so I need to put the disclaimer slide, but I think we can pass. So I'm the first in a series of 3 companies presenting to you today applications. What we share in common with my colleagues, Microbiotica and Ferring, is that we are all in the field of biologics, and we use the microbiome as a target because we want to change the microbiome based on what we have learned with the 2 previous presentations, for example. But also we use the microbiome as a source of pharmaceutical. So we are getting our material from from the gut microbiome initially, and then there's different level of processing, but that's the microbiome in the sense of its target and also the source of the pharmaceutical we are developing. The positioning of MaaT Pharma on that setting is that we want to leverage on the concept of the full complexity. So we are working with ecosystems that are full ecosystem, which means 250 species, to give you an idea. So it's very different to the probiotic you are buying in a pharmacy. And the density of the product is very huge. The overall concept is really to provide a full menu to the patient, and the patient will make his own selection depending on the kind of microbiome he has before the administration and also the status of his immune system. We are also positioned in oncology, and when it comes to microbiome and oncology, MaaT Pharma is the most advanced microbiome company in the world because we are in phase 3. We are treating patients as we speak today in a phase 3 in Europe with already 6 countries where we have centers recruiting patients in a disease I'll introduce just after. And we have established a proof of concept that modulating the microbiome is also restoring the functioning of the immune system. So in terms of the pipeline, we have 2 first products that are on the field of HSCT, stem cell transplantation. So we have one program which is to deal with the rejection of the stem cell transplantation. That's the first program, MaaT13 in graft versus host disease, phase 3, recruiting. Results come by the end of 23 to give you indications. And we have another program where we want to improve the survival of those patients going through stem cell transplantation before they develop complications. So just to give you an idea, the survival of those patients will be like 65% at 1 year. So we want to increase that level. So that's the 2 first indications in hematological malignancies. We are also evaluating our lead product, MaaT13, in combination with immune checkpoint inhibitors, as it has been explained before. And we have a new generation of products where we will not be developing the product from donors anymore, but from the banks, the master banks that we have created since a few years. In terms of data, the patients we are treating is in the phase 3. They have really a poor prognosis. Usually, this is a one-way ticket, unfortunately, and those patients, they're going to die for most of them. The survival at 1 year is around 25%. And here we have 2 sets of data showing that we have been able to inverse, to change the trajectory for those patients. The first set of data on the left-hand side is our phase 2 clinical trial, where we have changed the trajectory for 38% of the patients. We have also another set of data where it shows even a more promising signal because we have changed the trajectory for 58% of the patients. And that's coming from what we call the early access program, where we have seen that it's even more efficient when we treat the patient in a certain setting. And the phase 3 clinical trial that we are pursuing is exactly on that same setting as the one we have seen in the early access program. So that's with the MaaT13, the first product. The second product is a capsule. The first one is an enema. which is a high-density product. And the second one is a capsule. We have completed a phase 1B early this year, and we will be entering a phase 2B by the end of the year in any patients receiving stem cell transplantation. So with that, I will pass the microphone to Tim, which will present another modality.
Yeah, phase 3 in just 7 years. Congratulations, Hervé.
Yeah, exactly.
7 years since inception of the company. So we'll move to Microbiotica. Microbiotica, in addition to the results that Tim will present, is one of the most accurate and most powerful platform in the world that can actually map with a super high precision the composition of a sample. And in addition, Microbiotica has probably the best reference genome database, which is one of the key factors for actually progressing in that field.
Thanks, Isabelle, and it's a pleasure to be here. So, yeah, I guess we're the baby of the bunch. We're the newest company here, and we're just coming out of preclinical and moving into clinical trials. And what we're trying to do is develop precision microbiome therapeutics. And what we mean by that is we've heard all this fantastic evidence that the microbiome can affect the outcome of patients when they're taking especially immuno-oncology therapies. So the question we set out to answer is, well, which bacteria are the ones that are responsible for that effect? And if we can find those, we can make products out of just those bacteria. I could stop there, but I'll tell you a little bit more. This is our platform. As Isabelle kindly said, I think we like to think we've got one of the best microbiome profiling platforms. We're a young company, but the science has been around for a while. We were spun out of the Sanger Institute in Cambridge, which was one of the leaders of the within the Human Genome Project. And once it finished that, it moved on to other programs like the microbiome. So the platform has come out of over 10 years of research in building a huge database of gut bacteria, physically mapping their genomes, and that allows us to identify a bacteria, we believe, with unprecedented precision. So we start, as Isabelle said, and like everybody else, with clinical data. So we've run our own melanoma study in Cambridge. And the two men there may represent two groups of melanoma patients, one of whom will respond to checkpoint inhibitors, one who doesn't. And we can look at the microbiome with our precision platform and identify signatures of bacteria, small groups of bacteria that are associated with response or failure to those products. We believe we can identify some bacteria which others have missed. So those bacteria can be a biomarker. They can predict whether patients are going to respond or not to their checkpoint inhibitor before they take it. So that's useful as a biomarker in itself. But what we found is a small group of gut bacteria which is significantly raised in abundance in responding patients. So we thought, well, maybe we should think about putting those bacteria back in and seeing whether we could do exactly what's been shown with the FMT studies, where you take FMT from donor patients, from responding patients to non-responding patients, and switch their response. Could we find those bacteria which are responsible for that? So our own study in Cambridge, we found a small group of 9 bacteria that were associated with response across all of the patients. We then imported the sequence data from 4 other studies, 3 in the US, 1 here in Paris, and looked at their sequence data, and we found the same signature. So the same 9 bacteria were present in the studies that others had conducted. So that made us quite excited. So we've got the first universal signature for response. So we've grown up these bacteria. We can ferment them. We can grow them as pharmaceutical products. We've taken them through preclinical validation now. So we put them into mouse models of cancer. And when you give them along with a checkpoint inhibitor, we show increased tumor shrinkage and increased survival in those mice. We're now looking at some mechanistic studies to understand how those bacteria interact with the host cells and the immune system to promote that checkpoint inhibitor response. So that's our first product. It's moving into the clinic now. We are producing that. We're manufacturing these bugs. It'd be an entirely manufactured product. The bacteria are fermented. They're freeze-dried. They're encapsulated for oral delivery. So these bacteria were found they're found to be lacking in patients who don't respond to checkpoint inhibitors. this is an advanced melanoma patient again in the first instance. So we're going back into a clinical trial next year in exactly that same group. That's the precision medicine angle that we're pursuing. So we're going to be treating patients who don't respond to checkpoint inhibitors with advanced melanoma and giving these bacteria and seeing if we can replicate the FMT studies that have been done in Pittsburgh and Israel in the last couple of years. Melanoma is the start. We've got ongoing studies in non-small cell lung cancer, renal cell carcinoma. So we'll be looking for other signatures so we can expand our immuno-oncology portfolio. This is a big opportunity. It's a huge area in the pharmaceutical world. The immuno-oncology market is forecast to be the largest in the sector. But as we heard earlier, only relatively few patients respond. It's at best, with melanoma, 40%, 25% in non-small cell lung cancer, 10% in renal cell carcinoma. So if we can increase those response rates and give those patients long and durable cures, there's a huge opportunity both in terms of advancing medicine and obviously commercially as well. So looking forward to those results next year. So thanks for your time.
Thank you, Tim. We will now move to Ferring. So Ferring illustrates the pioneering and visionary European company acquiring an American startup and moving ahead, racing in this race of having the first product, microbiome therapy, likely to be approved in the coming months or weeks. And the big news 2 weeks ago was the positive recommendation of the Scientific College to the FDA.
Thank you, Isabelle. My name is Bilbo. It's a pleasure to be here today and represent Ferring Pharmaceuticals. And just very briefly on Ferring, our starting point, our core area is reproductive medicine. That is where we started more than 70 years ago. We're a privately held company and we're based out of Switzerland, and reproductive medicine is our core area. We are also strong in the area of gastroenterology and now have added microbiome therapies or are about to do that in this space. And the last area of focus areas of ours is urology. We are particularly also looking into uro-oncology, which is a whole new separate pathway, which is also shooting up. So if I can have the next slide. Sorry, that was... Here we are. So we started a journey more than 10 years ago, actually, exploring the microbiome. In the beginning, those were virgin steps, but we have sort of accelerated that. And that journey sort of accelerated with the acquisition of Rebiotics in 2018. And that led us on the path to do something serious with Clostridioides difficile, or C. diff among friends, an infectious disease which affects hundreds of thousands of patients, more than 500,000 in the US, and equally many in abroad. And that particular disease is what we have been focusing on, harnessing the value of microbiota-based live biotherapeutics in. This disease is seriously debilitating for the people affected by it. And what have motivated us the most over this journey have been listening to the people who have been part of the clinical program and have been positively affected, coming out of years and years of a vicious cycle where antibiotics essentially destroyed their gut microbiome, only to see that it was possible to do something about that. And that is what this investigational medicine, I should emphasize, it is an investigational medicine, is doing at the moment, RBX 2660, which we are developing. And as was alluded to, we just 3 weeks ago passed what we at least consider a needle's eye in the establishment of live biotherapeutics as a therapeutic entity, also by having a deep conversation with the FDA and panelists at an advisory committee, and coming out of that with a positive vote. And now we are working intimately with the FDA towards an approval of RBX 2660. Again, I cannot overemphasize the element of what this disease does. But as we look to the area of microbiome treatments, there is a number of other areas where this plays a role. And Tim and Hervé just talked about that. And this has also sort of been what we have been doing. We need to unlock the potential of the human microbiome, and we need to do that together, working with academia, working across companies, and this is key to us. We took a strategic outset in the area and indication of C. diff, but we see a use of the microbiome-based therapies in other gastroenterology indications. We also see, and we're exploring that at the moment, in reproductive medicine, in the vaginal microbiome, where we also can see applications of microbiome-based therapies, and particularly also in immune oncology, which also is an area of interest of ours. We see that there's a first generation of products, and we're working intensely to develop the next generation, which will be donor-independent therapies, where we will get to a more scalable solution of microbiome-based therapies. But every area has to start one place. And that's what we've been doing. That's what all of us are working towards. There was an era of small molecules. The era of biologics is slightly shorter, but it started a long time ago also. But we believe jointly that the era of the microbiome-based therapies is here and it's now, and that's the opportunity that we're grasping for. Thank you.
Thank you. It seems that we have time for Q&As. Before I'm given the questions from the audience, I'll start with one of mine. Mohamad and Hassane, you're in contact with the global range of therapies and ideas that all the microbiome field is developing currently. And I mean, for example, we as investors have reviewed more than 700 companies developing microbiome therapies right now. But recently, just prior to the summer, in parallel to the success of the Ferring product, there was also bad buzz from failed clinical trials and setback from Finch and Kaleido in the US, so even more visible than others. Does this recent setback has changed your view and your expectations of the microbiome therapy and the time frame it could be put in place?
No, I think this is a very important and fair question. Obviously, I don't know all the details about why this specific trial has failed or so on. However, my feeling in general that, and you know, medicine obeys to the rule of fashion like everything, that people are always thinking that you are going to solve the problem with whatever given specific bacteria or targeted product or whatever. And I believe that we are fortunate to work with MaaT Pharma, for instance, where they are developing a pooled product, which in another word is allowing our patients who are included in these clinical trials to have access, I would say, to the largest diversity of microbiome they need. Because I think we need to be humble at this stage that although we are convinced that the microbiota is playing a very important role in disease pathophysiology, but also in the way our treatments can be further improved, except maybe in a specific situation where you have a single infection like C. diff. Actually, in all other cancer conditions or autoimmune diseases, we don't know yet that we can solve the problem with a given bacteria or species. And this is why I think at this stage we need to have the highest level of diversity
and richness. And this is why I believe we do have such really positive and important results, what we see already with the MaaT 13 or MaaT 33 products that were shown by Hervé.
Thank you. Hassane, any additional comment?
Yeah, so I've been in the field of cancer immunotherapy since 30 years, so meaning we're not going to have the magic bullet overnight, right? It's going to take time, persistence, improvement. So I think we have to have a long-term strategy. I think the primary data and the premises are very strong. Now, what will be the end product? This is an open question, right? It should be a cocktail. A cocktail is very elegant. But what should be in, I think it's a matter of trying to find out. And we have not yet the answer to this question. I think it will take time. It will take trial, and we need to be persistent. And the second point I want to make also is also we also need to be have some common sense here. We also need to understand that a subset of patients will benefit from that, right? Not all of them, right? So the goal will not only to better refine and what is the treatment or what goes in the cocktail, but also who are the patients who can benefit or not of that, right? And to specifically address this subset of patients with such treatment.
Thank you. Dear entrepreneurs, having paved the way, would you have advices for other microbiome companies?
Well, I think one of our learnings has been that it's exceptionally important to work intimately with regulators across and along the development, because there's a big difference between what is agreed at an early stage and what's required at an early stage and what's good enough, and what over time, as this area matures, as learnings gets improved upon, what is required at the end of the program. And that's one of our experiences, is that we've worked intimately with the Food and Drug Administration in the US, and it's fair to say that their advice in the early days were as immature as our own understanding in a way, and we have jointly together evolved our understanding. I think that is a key element for all of us trying to establish this new modality.
Team Hervé?
Well, maybe I would say it's good to come second and let Ferring do the heavy lifting with FDA first. It certainly is, whenever you're working in a new modality, there are challenges, starting up studies, finding people who can manufacture your products. This is a new modality. I mean, I know, Hervé, I think you're doing it yourself. So a lot of the infrastructure that you typically have when you're developing new medicine isn't available to you. I think that's changing. Microbiome is there are more companies, there are more providers. So I think use the network. It is a close network. There's not many of us doing this. We all know each other, and it's and in any new modality, there's going to be lots of different approaches, and we need to learn from each other. I think it's one of the most supportive micro environments within pharma and biotech that I've worked in, and that's very helpful. So if you want to come and join us, you'd be very welcome.
Yeah, so I won't repeat what I've said, my colleagues, regarding the regulatory connection, but I'll add the fact that recently I did a mapping of who are the microbiome that are into the clinic, and mostly in oncology. I ended up with a club of 100 microbiome from China to West Coast in the US, just to give you an idea of the network. And I would leverage on the together from Carl, because I think when you are developing your company, you should not try, at this stage, you should not try to do everything by yourself. Because when I started the company in 2014, even my CDMO, I needed to build my CDMO. I was close to build my own CRO because dealing with a collection of stools and everything is not that easy. Getting DNA extracted from stools may be ridiculous, but it's not that easy. So when I started in 2014, we needed to create like everything. My advice to advisors, to entrepreneurs would be put your business model on paper, try to see where you want to focus, and then look at the network, look at the entire network and ecosystem, because partnership and open innovation, I think, would be more efficient than just getting money from the investors and develop everything yourself.
Thank you. Coming back to the hospital world, what is the role of the patient associations in this field, and does it differ from the other field?
Yes, I think so, because, well, obviously, interestingly, the patients are extremely enthusiastic and positive about it. And actually, they're asking, to get their microbiota being modified, treated, corrected, etc., which is a bit more complicated, and this is why we do have very well-established frames, I would say, to develop these treatments, because we're talking about true drugs, and they have to follow some rules. However, what I'd like to emphasize that we are tackling here a sort of a disruptive medicine, something which is not in line with the historical way we think about a drug, etc. So Hervé mentioned that you have to invent, I would say, almost everything. But all of this should be done in a very well-established frame to secure patient safety, but also an accurate and fair evaluation of the treatment. And why I'm mentioning this, because from time to time you would hear, and you would go to Google and internet and YouTube, and they would say, oh, fecal microbiota transplantation, this is very easy. You just take a little bit of poop, you just eat them or inject them. It doesn't work like this. You don't treat an immunocompromised patient by just getting, some fecal material and giving it. This is why it's very important to highlight and to explain to the patient, to the families, that we are really talking about, I would call it personally, revolutionary new medicine. It's a new frontier, and like anything new, you face conservatism. But this is why we're here, I think, to talk about it and try to push, because I'm a strong believer.
Thank you. Hassane?
Yeah, so I fully agree with Ahmad. And I would also underline that practically, we have been working with fecal transplant to proof of principle, but to implement this in the clinic with FDA regulation and to go in the same direction of Ahmad, this is becoming very complex. We have to face a large number of tests in patient, in the donor, so meaning the possibility to go with a cocktail, right? Pills and cocktail, this will really ease the clinical application of such approach in the future.
Thank you. There are in the microbiome more than 700 new clinical trials worldwide per year, and more than 1,000 new patents filed per year on this field. So pretty active, and it's mainly pushed by the startup or small companies communities. But what are the big pharmas doing, and what is their current state of mind right now? Maybe Tim?
Sure. I think they're intrigued. They're not ready to go yet, but they're intrigued. I mean, we're starting the immuno-oncology studies that I spoke about earlier. And we've been speaking with the pharma companies about them giving us their immuno-oncology drugs because they're expensive. It would cost us a couple of £1 million to buy those drugs for those trials. They all want to give us their drugs. They all want to see the data. They're all really intrigued, but they're not ready to jump. Yeah, I think. So they're watching. This is a new modality, that there will be failures. There have been failures earlier this year. Not everything's going to work, so I think they're waiting to see. I was speaking with the head of business development for one of the top 5 pharmaceutical companies, who said exactly that. He said, we're really intrigued, we're not ready to move, but if you can make a 10% difference in response rate to immuno-oncology drugs, that product's worth billions. So they're watching us, I think, and they're waiting. Ferring obviously leading the way, but the other pharma, I think is watching, waiting.
Additional comments from any of you?
Yeah, I would see, because now that since 8 years, I'm trying to convince pharmaceutical companies, and indeed the big names within the pharmaceutical company, I would agree that they have exactly the same appreciation of the situation as Tim. But I see the mid-sized companies moving, and I think they are more willing to take risk, because we are also in a field where it's biologic. We all know that the future of medicine would potentially be in biologicals, and they see an opportunity, the mid-sized company, they see an opportunity, and we see that because ourselves, we have 2 phase 2, 1 phase 3, so now we have data showing that it works, and indeed it's more easy with the mid-sized company as compared to the bigger company, I have to say. So I see a sign that we are moving from the early adopters only, like Ferring or Takeda or others, to those ready to go to the next step. And that's starting with the mid-size mainly.
Yeah, and I can add that I think it's important for all of us working in this space to look carefully at the different indications we're getting into and assess what is the bar we need to jump, because some of the areas are more mature. There's I wouldn't say a saturation, but to Tim's point, some of the companies say, well, show me the money, show me what you can do. And that means that we need to set the bar quite high for what microbiome-based therapies can do. The world doesn't need minute changes in therapies. We need big things, and I think we can do that, but it's on us to show this to the bigger companies also.
Thank you. Well, I guess we're almost to the end of this session. So maybe a general question which serves as a conclusion is, should you have a magic stick, what do you think is essential to accelerate the field?
Well, a very complicated question, but still, I think until we have cured all patients, we need to try these new tools. And it was mentioned. Today, it is the era of immune therapy, and for me, it's about combining the whatever microbiota agent, modulating agent, with all the available treatments, because this is where we will be able to move forward the immune therapies into the next step.
Any additional comment from any of you?
No, I think we are all expecting a big transaction. That's really what we are looking for, because that will, I think, unleash the potential, from a business point of view.
And I think everybody believes now we can treat C. difficile. I mean, you've proved it. There's emerging evidence that you can treat other diseases with microbiome, whether it's whole ecosystems or specific bacteria, especially in inflammatory diseases and immuno-oncology. So I think once we define the mechanisms by which they work, I think you're going to see a real acceleration in product pipelines.
Yeah, I echo Tim's point. I think as good as all the work that has been done over the years is, I think we are still not understanding good enough the microbiome and what the individual strains are doing in different therapies. I think that means, to me, it sounds strange from a commercial person like me, I need more basic research, more basic understanding in this field. And I think if I could sort of jump forward, it would be if we had sort of a much better understanding there. I think that would make everybody more comfortable with what we're doing.
And the last word from Pittsburgh to conclude this session.
So, to go in the same direction, we definitely need more research, right? But basic and translational. So we have the possibility to test a promising approach in patients, and I think we should do that. As long as they are safe, we can do that. Patient, there's some medical need, and learn from that. I think that's, to me, that's the best way we're going to sort out and find out what is the best and more potent microbial approach, right? So translational research, that's the way to go.
Thank you very much. Thank you to this excellent panel and all the input of your own experience and your vision for the future. I hope that the session convinced all of you that this is a sector to watch.
Thank you very much indeed for such an inspiring roundtable. A round of applause, please.